In this question, it is given that a woman has a X-linked condition on one of her chromosomes. So this chromosome can be inherited by option A, only daughters. It is incorrect because if the woman carries an X-linked condition on one of her chromosomes, she can pass it to both sons and daughters and not just daughters. Option B, only sons. It is also incorrect because of the above stated reason. Option C, both sons and daughters. Now a woman has two X chromosomes and she can pass either one to her child. Sons inherit their X chromosome from their mother and Y chromosome from their father, and daughters inherit one X chromosome from each parent. So if the mother carries an X-linked condition on one of her chromosomes, she can pass that chromosome to both her sons and daughters. Thus, it is the correct option. Option D, only grandchildren. It is also incorrect because the X-linked condition is inherited directly from the woman to her children, not just to her grandchildren. In this question, you have to choose the option that represents the characteristics of inheritance of blood group in humans. A, dominance. It is correct because in the ABO blood group system, the alleles IA and IB are each dominant over the allele IO. For example, individuals with genotype IA-IO will express the blood group A, as AIA is dominant over the allele IO. B, codominance. It is also correct. The alleles IA and IB are codominant, meaning when both are present, both the alleles would be equally expressed. For example, individuals with IA-IB genotype have blood group AB. Multiple alleles. This is also correct because the ABO blood group system is controlled by alleles IA, IB and IO at a single gene locus. Thus, this is an example of multiple allelism because more than two alleles exist in the population for a particular trait. D, incomplete dominance. Now, this is incorrect because incomplete dominance or blending of inheritance refers to a situation where the heterozygous phenotype is an intermediate of the two homozygous phenotype. So this is not applicable to the ABO blood group system or IA, IB, IO alleles. E, polygenic inheritance. This is also incorrect because polygenic inheritance involves multiple genes contributing to a single trait. Blood group inheritance is controlled by a single gene with multiple alleles, not multiple genes. So it is not polygenic. So the correct answer is option B. In this question, you have to identify the wrongly matched pair. Option A, starch synthesis in P is to multiple alleles. Now this pair is wrongly matched because starch synthesis in peas is governed by a single gene with two alleles and not multiple alleles. Hence, it is the correct option. Option B, ABO blood grouping is to codominance. Now, this is the correct match because the ABO blood group system exhibit codominance where allele IA and IB are expressed equally in heterozygous individual, that is, person having blood group AB. Hence, it is incorrect option. Option C, T.H. Morgan is to linkage. Now, this is also a correct match because T.H. Morgan is definitely credited for discovering linkage, that is, the tendency of genes located close to one another on the same chromosome to be inherited together. Hence, it is incorrect option. Option D, exotype sex is to grasshopper. Now it is also correctly matched because grasshoppers exhibit the exotype of sex determination where females have two X chromosomes but males have only one X chromosome. Hence it is also incorrect option. In this question, you have to select the correct statement. Option A, Franklin Stahl coined the term linkage. It is incorrect because the term linkage was coined by Thomas Hunt Morgan and not Franklin Stahl. Franklin Stahl is known for the Michelson-Stahl experiment demonstrating semi-conservative DNA replication. Option B, Punnett square was developed by a British scientist. Now, the Punnett square was indeed developed by Reginald C. Punnett, a British geneticist, to predict the outcome of genetic crosses. Thus, it is the correct option. Option C, transduction was discovered by Ace Altman. It is also incorrect because transduction, that is a process where bacteriophages transfer genetic material between bacteria, was discovered by Zinder and Leatherburn. And this Sidney Altman is known for his work on RNA enzymes, that is ribozymes. Option D, spliceosomes takes part in translation. Now it is also incorrect because spliceosomes are involved in splicing during RNA processing, which occurs before translation. In this question, you have to identify the scientist who explained or stated that the frequency of recombination between gene pairs on the same chromosome is a measure of the distance between genes. Option A, T.H. Morgan. It is incorrect. Though Morgan is credited with the discovering of the concept of linkage in genes on the same chromosome and demonstrated that some genes are inherited together due to their proximity on the chromosome. But he did not directly explain the concept of recombination frequency as a measure of genetic distance. Option B, Gregor Johann Mendel. Now it is also incorrect. Though Gregor Johann Mendel is known as the father of genetics for his pioneering work on inheritance patterns through his experiments on pea plants, but this work did not involve chromosomes or recombination frequency, as he was himself not aware of the physical basis of inheritance at the chromosomal level. Option C, Alfred Sturtevant. Now, Alfred Sturtevant, who was a student of T.H. Morgan, developed the concept of using recombination frequency to measure the distance between genes present on the same chromosome. Now, he created the first genetic map based on recombination frequencies, and he established that the frequency of recombination is directly related to the physical distance between genes. Hence, this is the correct option. Option D, Sutton and Boveri. Sutton and Boveri proposed the chromosome theory of inheritance, which suggested that chromosomes carry genes and these chromosomes are the basis of inheritance. But they did not address the concept of recombination frequency as a measure of genetic distance. Hence, it is incorrect. In Snapdragon, a red flower was crossed with a white flower, and in F1 generation, pink flowers were obtained. So, when pink flowers were selved, the F2 generation showed white, red, and pink flowers. So, you have to choose the incorrect statement regarding this concept. Option A, this experiment does not follow the principle of dominance. Now this statement is correct because incomplete dominance is observed in this experiment, and in incomplete dominance, neither allele is completely dominant, resulting in an intermediate phenotype like pink flowers in the F1 generation. Hence, this is incorrect option. Option B, pink color in F1 is due to incomplete dominance. Now, this is also true. The pink color in F1 results from incomplete dominance, where the red and white alleles blend to produce a pink phenotype. Hence, it is incorrect option. Option C, ratio of F2 is 1 red, 2 pink and 1 white. This is also correct. In incomplete dominance, the selfing of F1 generation, that is pink flowers, yields an F2 generation with a 1:2:1 phenotypic ratio, that is 1 red, 2 pink and 1 white. Hence, it is incorrect option. Option D, law of segregation does not apply in this experiment. Now, this is false. The law of segregation does apply in this experiment, as the alleles segregate independently during gamete formation, resulting in the observed F2 generation ratio 1:2:1. Now, incomplete dominance does not violate the law of segregation. It only affects the expression of alleles in the heterozygous state. Hence, this is correct option. What map unit, centimorgan, is adopted in the construction of genetic maps? Now, a map unit, which is also known as centimorgan, is a unit of measurement which is used in the genetics to represent the genetic distance between two loci on a chromosome. And one map unit is equivalent to a recombination frequency of 1%, meaning that there is 1% chance of crossing over occurring between two genes during meiosis. Hence, the correct answer is option C. In this question, you have to identify the condition when there will be no change in the reading frame of the given mRNA. Option A, deletion of GGU from 7th, 8th and 9th positions. Now removing 3 nucleotides, that is a complete codon, does not alter the reading frame of the mRNA. And the genetic code is read in groups of 3 nucleotides. So if a complete codon is deleted, the downstream codons remain unchanged and the reading frame is preserved. Hence, this is the correct option. In this question, you have to identify the nature of variations caused by mutations as proposed by Hugo de Vries. Option A, random and directional. It is incorrect. Mutations are random changes in the genetic material, but they do not occur in a way that leads to a specific direction. Option B, random and directionless. Now, mutations indeed occur randomly without any specific aim or purpose. They are directionless, meaning they do not occur to benefit or to harm an organism. And Hugo de Vries proposed that mutations are spontaneous and can lead to either beneficial, harmful, or neutral variations. Hence, this is the correct option. Option C, small and directional. Now, it is also incorrect. Now, mutations can be small or large, but the term directional suggests a predictable or advantageous thing, which is not correct. Option D, small and directionless. It is also incorrect. While some mutations can be small, but they are also not always so, and mutations can lead to significant genetic changes. So it is incorrect to generalize them as large and small. the term directionless is correct. In this question, you have to identify the incorrect or the false statement. Option A, male fruit fly is heterogametic. Now, this is true. Male fruit flies, that is Drosophila, are indeed heterogametic. That is, males have two different, that is hetero, type of sex chromosomes, X and Y. Hence, it is incorrect option. Option B, in male grasshoppers, 50% of sperms have no sex chromosome. Now, this is also true. Male grasshoppers have an exotype of sex determination system, where males have only one X chromosome and no Y chromosome. So, half of the sperm carry an X chromosome and the other half carry no sex chromosome. Hence, this is also incorrect option. Option C, in domesticated fowls, sex of progeny depends on the type of sperm rather than egg. Now, this is false. In birds, including domesticated fowls, females are heterogametic, carrying ZW chromosome, and males are homogametic, carrying ZZ chromosome. So, the sex of the progeny is determined by the type of egg rather than the sperm. Hence, this is the correct option. Option D, human males have one of their sex chromosomes much shorter than the other. This is also true. In humans, males have one X chromosome which is longer and one shorter Y chromosome. Thus, it is incorrect option.